The goal here is a discussion. So we really want to just have an open dialogue. We've heard a lot of different kinds of people here at AFTD's education conference. So any good discussion is first prefaced with awareness of who's in the room. And so I wanted to just kick us off by taking a quick moment. I know people are still coming in, so things will evolve undoubtedly as more people come in. But who do we have in the room? So I have to ask, if you are from a family that is impacted by FTD, can you please raise your hand? Thank you. If you're a person diagnosed, you yourself are diagnosed, can you please raise your hand? Thank you. Have you at some point now or in the past taken care of somebody with FTD as their partner, as their caregiver? Can you please raise your hand? Thank you. Are you the child of somebody with FTD? Thank you. Are you with a company that is working to develop treatments for FTD? Can you please raise your hand? And we've asked most of these folks to sit in one side so that the microphone, as questions come up, can also go to them relatively easily. Are you an academic researcher working on FTD? Smaller number, but we do have quite a few over there. Sounds great. Are you a genetic counselor? All right, got a few over there. Are you a different kind of healthcare provider? Hey, you're a healthcare provider, aren't you? Emma, Dr. Rogalski? There we go. Thank you. Are you an AFTD staff member? Perfect, thank you. Are you an AFTD board member, ambassador, or other volunteer? Thank you. And last question, are you here working with a different advocacy organization, either with whether you're, some people volunteer both with AFTD and other organizations. Are you here with a different advocacy organization? There's one. Oh, I'm surprised there aren't more. Well, what's essential to remember is that treatments for FTD are only going to come about when all of us are working together. So thank you all for being here in this room. So before this session started, some of our team members asked questions to the community or solicited questions from the community. So we have one or two just to get us warmed up and get the conversation started. But again, the goal here is a discussion. So please think about what questions you would like to ask. And then we're going to have some colleagues running around with a microphone to try to help you ask them live. And then I may not be the person who's the best expert to answer the question, which is why I'm so grateful that we have lots of different experts in this room, and we may be having them answer the question. So we came off of a session before the break that had 6 different clinical trials, all focused on progranulin. So one of the most common questions that we hear from the community is, if you are from a progranulin family, how do you decide which trial is right for you? And I think it's a really important question. We've heard different parameters matter to different people. So there are some people where placebo is just not something they're willing to look at. So they're wanting to look at gene therapy. There are other people who don't like the invasiveness of gene therapy. There's also differences in every trial and how far do you have to go? How close is it to your own home? There's questions around how frequently will you have to go into the clinic? And there's differences in inclusion-exclusion criteria, meaning will you be eligible? It does vary across trial. So what we often like to tell people is take a look at each of those different trials and see which one might fit you. But the other thing we often say is please speak to your healthcare provider and they can help you make that decision. So I'm going to ask Dr. Rogalski as well as Dr. McMillan over here. Amanda, if you could ask Dr. Rogalski first, and then Dr. McMillan, if you wouldn't mind, how would you answer the question?
Thanks, Penny. I appreciate the opportunity to talk with you all today. I think you hit on a lot of the major points. This is a deeply personal decision, and so taking your time to figure out what the right fit might be for you, and it might not be this set of trials for any number of reasons. Could be exclusion, inclusion criteria, could be that it's not close enough to your home, but really understanding and making that decision jointly with your family, with your healthcare provider, and understanding, this is really an exciting time. So I was just, marking at the registration table. I remember a time when we didn't even know the third family of diseases that caused FTD. We called them DLDH, dementia lacking distinctive histopathology. That meant we knew it was different than the other pathologies, but we didn't quite know what the bad protein was. We now know these bad proteins, and we now know some of the genes that are causing them. So we're in a really unique and exciting time. So please take the time to consider if it might be right for you. Hopefully that's helpful.
Thank you. Dr. McMillan?
I don't have a lot to add. I think you guys have both answered the question great. And just to re-emphasize that it really is an individualized decision. And there are a variety of things to consider that range from the risk factors involved in a trial, that range from what information you might learn in the trial. So for example, some trials you might not have to know your genetic status, and other trials you might have to know your genetic status, and things like that to consider. So it's a very holistic perspective, and being as informed as possible of what information you can get from your clinicians is an important discussion to have.
Thank you. I will also flag that the Progranulin Navigator is a different nonprofit's resource that might also be helpful to families. So a second question that we have often heard is, why all the progranulin and what about the rest of us? Right? So I think that there's a lot of different complex reasons for that as well that have to do with the biology of progranulin, of whether or not doctors think that they're more likely to be able to successfully treat it. I will be blunt that a given clinical trial has a really high % failure rate, meaning any company investing in drug development has a high chance that a single type approach won't work. And it's a really hard fact to come to recognize. It's why AFTD is committed to having more shots on goal, to making a landscape that it's easier to test treatments in all types of FTD. But for a drug development landscape, some of the progranulin components of biology are thought to be easier or higher chance of success. But I think there's also aspects where the science is also just more mature. But I'm actually going to punt this to some of the companies working in this space. Some of you are also working on C9orf72, for example. Some of you are at companies that might also be working on Alzheimer's and looking at other aspects of tau biology, for example. Do we have any volunteers? Dr. David Cooper from Aviato Bio.
Thanks, Penny. Happy to start the discussion. So I think, as you've said, the biology of GRN seems a bit more straightforward. It's a protein, it's secreted out of cells, so it can transport, and it makes sense to use that as a first target. Our approach has been really, we look at each type of disorder, we look at what exactly is the genetics and what's the underlying mechanism. So the way we've looked at FTD, ALS, Alzheimer's and other tauopathies is we look at it in broad buckets. FTD-GRN and GRN replacement therapy seems to make the most sense. In terms of tau and MAPT specific mutations, tau itself accounts for about half of all FTD. So our approach is we're working on a tau treatment that can be used for not only MAPT specific genetic mutations, but tauopathies like Alzheimer's, like PSP, like CBS. So we're really trying to do something on a broader scale there. In terms of C9, I was at Unicure before this, and we had several versions of a C9 treatment. It's much more complicated biology is probably an understatement. But it's part of TDP-43 proteinopathies, which GRN is also a part of. So we've tried out several mechanisms already, and we're continuing to look at how do we target that biology in the best way that gives a likelihood of success so that we could bring it to clinic, as well as the potential that it can translate then to broader populations from whoever we first study it in. So I think a lot of us really try and take that kind of approach. Sometimes one treatment gets ahead of the other in our animal studies or in our development. And we certainly take many passes at all of them as we try and bring treatments forward. So what you see in clinic is the result of often 10 plus years of work to ever get a generation of product out there. But hang in there, we are working on all of it. So I don't know who else wants to.
Hi there, Paul from Vesper Bio. So I'm going to say, yeah, we're in FTD GRN right now. We're not saying no to the other types of FTD. It's just not yet. I think you've just said a lot of the great things. It does take time. You've also acknowledged that we do have to answer to the people who pay the money to run the trials, unfortunately, and give them the best shot on goal and take things in a stepwise approach. The first thing we have to do is to show that we have something that's safe and that we're not going to cause more harm than good. And once we've done that and we've shown there's a reason for optimism, then there's a great reason to push into other FTD types, subtypes, but it does just take that time. I have to apologize, but that we really feel the pull from your side, and I have to acknowledge the great work that you are doing to really pull the community together.
Thank you.
We hear it, so thank you.
And Stella, would you mind actually coming up? It'll make it people, Stella McCaughey from Elector, and I believe Dr. Richard Tsai from Denali was also going to answer. I think it'll help just if the companies can be in the same region. Oh, thank you.
Hey, Will Chu from Passage. So we are currently studying both FTD GRN and FTD C9, and I want to use that as an example of things can move forward. So we started in FTD GRN for all the reasons that have already been elucidated, which is it is the highest probability of success because we understand the mechanism the most. But because we've done enough work in our current asset where we understand that we can administer it safely and we understand that it does what it's supposed to do, now we've seen there's enough preliminary evidence that we want to try it in FTD C9 because patients don't really have any options. They don't have enough options right now for research, patients with FTD C9. So it's just an example that the words that you've heard, they actually do happen. We all do expand over time.
Richard Tsai from Denali. So I think what everyone said about the biological understanding and the science and having to have a deep confidence in your drug that works against the disease's biology and has safety and potential benefits is absolutely true, and it is the major factor. But I just want to also take a step back and say that there are other things under consideration as well. There's a lot of companies here doing FTD progranulin clinical trials, but if you ask any of us, how easy was it to run that study? How easy was it to design that study? I don't think any of us here would say, oh yeah, it was great. It was so easy. So, I think FTD, of course, still a fairly new disease, and maybe I'll take a step back. So some people are probably wondering, oh gosh, there's so many companies doing Alzheimer's treatment. They have drugs against tau, drugs against amyloid. Why not FTD? And it really is. FTD is a newer disease. We're still learning a ton about it. There's progress all the time, but it's not quite as mature there in terms of where to find the patients, how to measure the disease progression. And it's even more challenging because FTD is more rare. So, just the efforts here are exactly what will make the companies more interested in developing a drug and a clinical trial once they have that biology understood. So, something like the Cure MAPT FTD is exactly what's needed to help the companies realize there's enough patients out there's enough interest out there, and as you guys build these cohorts and help the researchers like Corey and them to learn about how your disease progresses, what's the best way to measure them, and maybe that will create this sort of momentum and energy for the companies to sort of band together and be like, yes, this is going to be like Alzheimer's, we can do it together, and so.
So I totally agree with everything Richard just said, but I just wanted to share that at Elector, we are also studying the elevation of progranulin in Alzheimer's disease. So it's a similar molecule, but different than the one I presented. And so we're really looking forward to the results of that study, but really, as I mentioned, we're looking forward to the results of this FTD-GRN study in latozinemab, because we'll get answers from that, right? We all hope the answers are positive and that it worked in the patients that we tested it in, but we're gonna learn. And those learnings can be applied to other types of FTD and other types of neurodegenerative diseases. So I wanna just reiterate what Richard said. I think the efforts of the AFTD, of the AFTD Disorders Registry, of the different Cure MAPT, Cure GRN, being a part of those does make it easier. I've been at Elector for 5 years, and I can tell you that recruiting this trial was not easy. I lived it for 5 years. And so being a part of this community and finding that out so that we can quickly assess where the patients are and the feasibility of it is really important. And so I think working with the AFTD, the registry, and the Cure groups is really important for that. So.
I think another important thing to recognize is just because your gene isn't in a trial doesn't mean we're not learning important things that are going to inform the genetic mutations that you might have. And a great example of that is in prevention trials in ALS, where SOD1, there's now a FDA-approved therapy for SOD1 for ALS, which now is being modeled in a prevention trial. And we're hoping to use information that we're learning from designing that trial to design prevention FTD trials. So even in a different disease, the information we're learning is helping you, even though that there might not be an immediate trial for you to participate in.
Those are some wonderful answers. I'll also add, there's work happening on every single type of cause of FTD. The question is, where has the science evolved? And where do the companies have enough confidence that they have an approach that's likely to work and that they'll be able to find enough participants for their study? Numbers have power. And so I think that is central to why we've got the registry. I think it is the power of groups like Cure MAPT FTD and Cure GRN and Cure VCP as well. And I also want to call out ALL-FTD, and I'm going to ask Leah Forsberg over there, because ALL-FTD is doing a lot of work on not just the 3 most common genetic causes and not just the sporadic forms. Dr. Forsberg?
I'll get up too. Hi everyone, I'm Leah Forsberg. I am the ALL-FTD representative here today. If you've not heard of ALL-FTD, we are the natural history study for FTD in North America. We have 28 sites across the United States, 2 of which are actually in Canada. And we are just so grateful, first and foremost, for anybody who's participated with us, either early days when we were 2 separate studies, Artful and Lefties, or if you've continued with us or just recently found us and are hoping to get to join the group. We are at sort of an in-between period for those that are aware or may not be aware. NIH funding usually comes in 5-year increments. ALL-FTD had the fortune or misfortune of getting off the ground in March 2020, which, if you remember, that's also when the COVID pandemic hit. Because of that, we actually were able to do an extra year in the middle of our study. So we've had 6 years of funding, truly 5 years of activities, but it does mean that things are winding down as we are nearing the end of that sort of initial NIH grant period, we have submitted for our renewal application. For those that are unaware, this process takes time. So we are hopeful that we will be funded, and we will hear about that soon. We are very optimistic. We received a really good score when it was reviewed scientifically. So if you're also unfamiliar with the process, you submit a renewal grant, it's reviewed scientifically, and then it's reviewed again to be considered for funding. So multi-steps, we're almost there, but we're really hopeful and remain optimistic. We, as Penny mentioned, have individuals who are from families with known genetic mutations are enrolled. We also enroll people that have a strong family history. So whether the genetic mutation is unknown because the person in your family with symptoms has been tested, but no mutation has been found, or you have a strong family history, but no one who had symptoms was ever tested, so it's not known what mutation exists in your family, you're welcome to join. In all FTD, we take all comers from families, so that means if you know your status, and if you know your status and you're negative, we welcome everyone at this time. We also are thrilled that we have a very large sporadic cohort within our study as well, because we know, FTD is very heterogeneous, and it is important to have everybody represented in the group, hence the name All FTD. Any other questions I can answer, Penny?
I think we're good.
Okay.
Thank you, Leah. All right, so we're gonna, if you have a question that you'd like to ask in general about research, raise your hand. We've got one right there, but we have a question ready, and then next to over there.
Thank you, Penny, for organizing this awesome opportunity. It kind of answered a little bit of my question, but it's focused on the less common forms of genetic FTD and what's required in order to gain interest and reduce risk in the eyes of biopharma to encourage investment into these less common, more biologically difficult pathologies. So my question is, what is the most valuable thing that would help you to invest in these smaller genes? Are there specific things that you need? And Penny, you already kind of mentioned some of those things.
And I think your question is really for the companies in the room, as I'm understanding it, right? So would a company like to? All right, we have a volunteer.
Me again. So not just CEO of Vesper, I'm also a part-time investor, so maybe I can
Perfect.
help give some nuance to that. When we look at investments into new biotech companies, we really want to understand two things: the technical risk and the commercial risk. What does that really mean? How strong is the science? And that comes out of universities. The foundation that I work for donates around $140 million a year to Danish research. And out of that comes, along with all the other funded research in Denmark and around the world, excellent science that is maturing the whole time, as you've already said. And at the same time, the commercial potential is being developed as we learn more about the prevalence, the number of patients, how their disease progresses, what biomarkers are being developed to allow us to follow disease progression, and how much confidence we have that a treatment may actually have a beneficial effect on a disease trajectory. Now, in our case, we can invest into, or we do invest into, very risky projects already just coming off the lab bench from some professor, or we can go into very late stage clinical trials and invest into those. It's really everything combined. Is there an unmet need? Conferences like this make us understand that absolutely there is. Mm-hmm. Is there real science there? And we can do a lot of diligence on that. And is there a willingness from people out in the regulatory field? Does the FDA support new approaches to developing drugs? Yes, they're doing that over time. And are there payers out there willing to help us develop a commercial potential, even if it's a smaller number of patients? And so, yes, all combined, that can come together as we get more science, as we get more understanding on the regulatory side. But it's hard to put an exact number or an exact process on that. It actually becomes more a feeling, when is it enough? When is there a conviction? That would be my insincere answer, I suppose.
I think I can also answer where, frankly, a lot of AFTD strategy is all designed on how can we make this the space where drug development wants to focus. And there's a lot of different tactics that we use, conveying the urgency of the need. Things like the patient-focused drug development meeting where the FDA understands. I mean, I have had FDA folks tell me about how during that meeting, there were FDA staff crying, communicating to each other. It is powerful to hear directly from affected families. And it really makes a difference and it can move people. But that's one small piece, right? Because there's a lot of horrible, horrible diseases that are out there. And whether it's the investor or whether it's the scientist, they want their work to make a difference. So where is it that they can test their concept, their ideas? So there's things related to the All-FTD natural history study to be able to understand how these diseases progress. There's tools to improve access to diagnosis, and indeed all of our work about medical education, the diagnostic checklist, the work you all do in the communities where you go home to raise awareness of FTD, all of these things help to increase the numbers of people that are out there. The FTD Disorders Registry, the whole concept, numbers have power. How do we characterize people with a low-cost tool wherever they happen to be so that when there are opportunities, we can also help companies find where in the country or where in the world might people be and characterize the population. I mean, it's developing the tools and the outcome measures, but I did not give the other companies a chance to respond, so please.
Oh, well, mine's more about the science at Stella again. And that's that when you think about FTD, I don't think any company wants to just be in one small fraction of FTD, right? It's that's where the science has taken us based on the mechanism of what we're studying. But as many of you I'm sure know, and for those of you that don't, there's different underlying pathologies, and that's true in all neurodegenerative diseases. But for instance, in FTD, if you know that your mutation or your disease is TDP43, and right, if they know that, right, because we don't have a good marker to track that live now, but then the science can follow that and say, like, if a drug has an impact on TDP43, then one can reasonably estimate, right? And so drug makers can look at that. If they know that it's MAPT and the science says that, you know, it might have an effect on tau pathology, or if another disease, you know, another mutation has that, then the science can follow that. And so that doesn't answer the financial question, but the science can follow that. And, you know, I'm sure all drug makers would prefer to be able to have a drug that targeted more people and had an impact on a broader population.
Question or answer? All right, was there anybody, I think we had a question over here first, and then over here to the left. In the middle first, please.
Sure. So I've got a part of a two-part question kind of on the same thing. So if you can stop TDP-43, will that collapse the C9 gene? And then on the inverse, if you collapse the C9 gene, will that fix the TDP-43 issue?
Dr. McMillan, would you like to answer that question?
Would you like to do a PhD in my lab? Because it's a great question. The short answer is we don't know. The complexity, the more we learn about the diseases, the more complex they get, which in some ways is good because we're getting closer to solutions, but it also opens up splitting things into lots of different subgroups. So we talk about TDP-43 broadly, which accounts for half of FTD and about 99% of ALS. But when we look at TDP closer in the microscope, we see that there's actually different subtypes of TDP. And now we're up to type E. So we have A, B, C, D, and E. And there's other subtypes of TDP that we don't even know how to classify. So we don't know whether gross TDP-43 therapy will take care of all types of TDP. But we are learning more about those subtypes, which are helping us drive the biology. So we recently identified that TDP type C, which is the most common cause of semantic variant primary progressive aphasia, is tethered with another protein called annexin 11. And that really opens up a whole nother opportunity that maybe there's some molecular basis that's driving these TDP subtypes. And maybe it's not even TDP that's the problem, and maybe it's these other things that are attached to it. So the closer we look in the microscope, the more we learn, and we'll, I guess another example of this is in the case of Alzheimer's disease, where we used to make the case that studying FTD was a simpler model than Alzheimer's disease because half of people with FTD have tau pathology, and everyone with Alzheimer's disease has tau pathology, but they also have amyloid pathology. It's a dual pathology. And in biomarker discovery, a giant lesson we learned is when we developed PET tau tracers to image tau in the brain, the idea was these tools were developed under a microscope to bind to tau pathology in Alzheimer's disease because it was more prevalent and easier to do in the lab. But then when we studied these PET tracers in FTD patients, it turned out it doesn't work because the tau in FTD is a little bit different from the tau in Alzheimer's disease. So we'll learn through trial and error whether we can lump or have to split into these smaller trials, but this is kind of the motivation behind what we talk about precision medicine or personalized medicine, where we do think ultimately we want to tailor treatments to individuals.
I'll just also mention that one of the interesting things on the TDP-43 side that I think is going to bring a lot more resources to the space is that about 30% of people with Alzheimer's also have a TDP-43 pathology. And I think the Alzheimer's space is increasingly paying attention to that. It's also the pathology that tends to underlie or always underlies ALS. So I think all of these things is part of where, it's complex, but I think there's also other sources of investment that are going to be coming in that are going to be then things that we can leverage for FTD therapy development. You had a question? Yes, you go to Mike.
Yes, I do. Okay, so I know we're all talking about genetics, but I'm going to dumb it down and take genetics out of it for a moment. For those of us that are here that may be kind of over our head, how do we ensure that PET scans, brain MRIs, and all the progression of this terrible disease gets to the hands that it needs to, whether that be researchers, or doctors, just to help even get before the genetic part of it?
So am I understanding your question correctly, that it is, as these tools get developed, how do they get in the hands of the healthcare providers that are working with families? Is that?
Essentially, yes, but as we do the testing, is there places we should be turning in those results just to see the progression of the disease before we get into the genetic part of it, that might make a difference, like PET scans, imagings, all those things?
So healthcare and research are very distinct. So if you're not enrolled in like an actual clinical research study that you've consented to, then it's true that medical records do sometimes get mined for a lot of different research questions. They usually don't have the ability to go to the individual scans in the same way. I welcome being corrected, by the way, if anybody is going to disagree with anything I'm saying. That's frankly part of why the registry was also created, because it's not a place that you can upload your individual PET scans, but it is a place where anyone can go if you're able to consent to the research study in the United States or Canada, and you can upload your genetic test results. You can answer all sorts of questions. And we would prefer if everybody could get into all FTD, but that nobody can afford that. So it's this lower cost option to characterize the population, and you can answer questions. We don't have the ability to take your imaging results either. But frankly, maybe we should add in more questions for people to answer about what kinds of imaging have you gone through as part of your healthcare. Does anybody have another aspect of this answer? We have 2 volunteers, or 3 volunteers, my goodness.
I think one really important source of momentum that the field has really experienced that I think is very unique to the FTD field is that data sharing has become a really important part of our research efforts. And that happens through participating in consortia where we know, so if we were splitting TDP into lots of different subtypes, we might have 10 people of each subtype at the University of Pennsylvania where I am, but that's not enough to really understand the complexity of disease. But if every FTD center in the country takes their 10 people and pull it together, then we have 200 people, and then we can start to really answer the questions to do that. And in the FTD field, there really is a small scientific community that work really closely together that's very unique. And I work in several other diseases as well, and it's part of the reason why I love doing the FTD research that I do is because of that camaraderie of the groups and sharing. So participating in research is important, and one thing that individuals can do beyond sharing their data is advocating on the importance of data sharing and making it known that you want your data shared when you consent in research studies. And organizations that fund research should require data to be shared and encouraging people to do that. And likewise, pharma should make data available to scientific researchers so that they can do secondary analyses of the studies so that we can, even if a trial fails, we can learn a lot from that failed trial to inform the next trial.
Dr. Rogowski?
Yeah, so maybe just to add on that, yeah, the bringing of people together, of researchers together is actively happening. It's how part of all FTD is so successful. There's been another consortium that Corey and I have also been involved with in addition to LFTD. But you can ask for your medical records, you can ask for those scans, and researchers may be able to ingest them into their research studies. Another aspect that's really growing is if you get a scan at one place, it might use a different scanner that would be slightly different than what we would use for research, or they might have timing that's a little bit different. We're working on harmonization methods so that we can ingest different types of data, but still glean really important things from them. So it's not lost, and I think as we improve our methods, and I think this is to Penny's point, there's research happening at all of these levels, and they all eventually come together to help. The rising tides lift all boats. So I think ask for your data and see if there are studies that are willing to take it. If they aren't today, there may be one in the future. And some of these help with the outcome measures. So if we can't measure TDP from blood yet, or through a PET brain scan, these are also in development, and these will make a difference when we're thinking about pharmacological options so that we know that there's target engagement and change in the thing that we are, in the diseases that we're trying to target.
One thing I forgot to mention two things. One is that if you're going through healthcare and your doctor's not mentioning research, you can ask. A lot of times there's different centers and you may not be getting seen by a clinician who's specializing in research, but that researcher might be across the hall. And so there may be opportunities for you to participate within the research center, within your clinical care center. And then the other piece is that if you are a participant in the FTD Disorders Registry in the United States, you can allow access to your medical records so that things like what was your diagnostic journey, what were the clinical tests that you went through, what were the diagnostic tests in general. There's a lot of information within there that can be collected just as part of your clinical care, and you just need to grant permission to us to have that data ported in. Any other? Dr. Cooper, and then I think we have a couple other questions.
Thanks. Just quickly, MRI is one of my bully pulpits, so I'll just say a couple words. I think, as a neurosurgeon by training, who looks at your scan is critically important, right? And we hear stories every day about the challenges in the diagnostic journey. What galls me is when someone's had an MRI and it's misinterpreted, it's not looked at quantitatively, et cetera. In trials, we certainly use very sophisticated AI to quantify different brain regions, et cetera. It's one of the benefits of all FTD and GenFi is that there is an ability to go with new algorithms, new AI to say, how can we define based on hundreds of scans what exactly disease progression looks like, and is there a way to make it essentially foolproof so that when you go in and you go on a Siemens scanner at whatever hospital, they should be able to push a button, that gives you a predictive score that this is Alzheimer's, this is FTD, et cetera. And those things are published. It's just they're not on GE scanners and Philips scanners and Siemens scanners, versus if you have a cancer, there's radiation oncology packs that will tell you the size of any tumor in the body built into almost every scanner. So it's just not there yet. So there's huge value to getting multiple scans in a research registry like AllFTD or GenFi, where that allows us to be able to look at that stuff. But it's also an opportunity when you guys have had experiences with these long journeys and misread scans to go to your institutions and say, is there a way within the network that you guys can do QC and basically look at scans of patients with dementia diagnoses, et cetera, and figure out if there's more patients out there and where's the balls being dropped. Because this should be something easy. Like all these images, even in a hospital network, are stored as DICOMs in a centrally available place. It should be very possible to go back and interrogate them and figure out who has characteristic MRI patterns. In Huntington's, there's so many tens of thousands of scans that they were able to go back and redefine stage 1 disease as being the onset of atrophy that's beyond what would be expected for your age. So it's not first movement disorder, it's the onset of atrophy. But they did that because there's tens of thousands of scans in research registries that enabled them to do that and look at it in a very sophisticated way. So use that MRI, leverage it.
Well, I certainly hope so. AFTD has an open call for proposals right now for researchers wanting to develop tools to improve access to diagnosis, and I hope that we get a proposal along the lines of what you're describing. Other questions? I'm not sure who is. Please, go ahead.
Thank you. With all of the great research and clinical trials going on, I don't think there's anything that has the promise of reversing the effects of FTD, especially the people that are very advanced in the disease at this point. My question is, when you're doing clinical trials, is there a certain age group or point where you're searching for participants? Because is it in 30s, 40s, whatever? Because if the symptoms begin probably around 40s or whatever, how do you then measure the results if you're having the effect of preventing the disease? Are you monitoring the symptoms, whether they continue to develop, or how are you determining the effect of the clinical trials based on the age of the participants?
That's a really great question. And I'm going to suggest, so I think we only have 5 more minutes, and I saw many other hands up. So I'm going to give a short answer, and just to try to get to some other questions as well. Every given company tries to design a clinical trial, and they're going to set for themselves what's the age range that they'll allow, as well as what's the range of disease severity that they would allow. And you'll note that the phase 3 clinical trial that Dr. McGaughey presented on a lecture, they had included pre-symptomatic people who were not manifesting the disease, but who had a high neurofilament level, meaning they had a biomarker that they thought might predict that these are individuals about to develop disease. If you have somebody who's 20 years away from developing disease, how do you know if the treatment's working? It's not, you have to have tools to measure if the treatment's working or the trial is just a waste of everybody's time and resources and the patient's time or the family member's time. So in FTD, you can have somebody who's 40 at late stages of disease, and you can have somebody who's 80 at early stages of disease. And so they rely on a lot of data from the AllFTD natural history study to try to project what is the likelihood of somebody progressing at a given rate, and then design their trial to say, how am I going to see if my treatment's going to work? And they include both usually the assessments of how is somebody functioning and feeling, as well as the biological measures to try to see what's happening within the brain. Next question.
Hi, I have a 2-part question. My first one is for the biopharma companies, and do they consider NFL the key biomarker for FTD? And my understanding is the FDA has not fully recognized NFL as a biomarker? And I've wondered if there's movement on that.
It's an incredibly smart question as well. So in the sake of time, I'm going to ask one company to answer that. Dr. McCaughey, since Elector's been a little bit further in their phase 3 and has tried this in different ways, would you like to answer?
I would say that we're following neurofilament, and we did use it in our pre-symptomatic individuals. However, I think that our trial will tell us a lot about neurofilament because we haven't really had a study that's shown us over a long period of time. Well, we've seen in natural history studies what neurofilament does, but we haven't seen like what an intervention could do and if it does impact neurofilament or not, right? So collectively, in a phase 3 trial, you'll be able to look at MRI and clinical outcomes as well as biomarkers like neurofilament and understand what the role is. But I think more is to come, and there's other great work going on. Maybe some of you are part of the neurofilament surveillance project. That's also doing great work to follow the role of neurofilament over time and give us more data so that we can interpret results. But
I'll just add that in the AFTD Research Roundtable, where we're working with different companies, the FDA, the EMA, and a lot of the academic research leaders, neurofilament is constantly getting raised. And one of the things that we hear is that the science of how confident are we in what it's telling us is a little bit farther ahead, say, for the ALS space compared to some of the FTD spaces. And it also varies across different genetic forms of FTD, which complicates then how can it be relied upon in a given clinical trial. It's part of why AFTD is invested, as many have, in that neurofilament surveillance program that Bluefield Project leads, as well as there's an initiative at the Foundation for the NIH, where we and others are also trying to advance the science of neurofilament, because I think it is by far the most promising tool right now.
Thank you for the efforts that are going into everything. And forgive me if I missed this part, but I understand that the overall is that you're trying to kind of replace the progranulin or whatever it is that is the protein that is at a loss. And that's something that you can measure, and I understand that. But as you're doing these trials, what behaviors and those kind of things are you seeing that are improved? Have you seen any? Are they starting to talk a little bit more? Are they able to function better? Are they executing a function more? I mean, to me, that's kind of the bottom line, obviously, is that we want to help our loved one function again.
It's absolutely the bottom line.
So
right?
Are there things that you've seen that they're doing as you're going through these trials, and not just measuring what you're replacing, but have you seen behavior changes and things like that?
And I think I will preface it with that, some of them may not be able to tell you, depending on legally what is the status of their company's trial of what they might be seeing. And I think that it's what we all want to see. And I know that one of the things that I draw the most hope from is that what I've heard from the tofersen studies in the ALS space, because it is the type of transformative treatment that we have been wanting, where we've heard reports that up to 50% of people have gotten better and not even just been stabilized. But that's tofersen within the ALS space. And I have not heard, would anybody
Oh, I was just going to say that, a placebo-controlled trial is completely blinded. So we have no idea who's on placebo or who's on the active drug. And that's really important because otherwise the data are biased. And so your question is what we all want. It's what we all hope for. It's why we all do the work that we do. But until the trials are read out, we don't know. In the case of open label studies, we've had open label studies at Elector, you have data, but you don't have a placebo control to compare it against. And so that makes that data harder to interpret in some cases. And so, but do you want to add anything to it? Oh yeah, but we 100% do measure clinical function. There's different scales. They're called clinical outcome measures. We measure, there's an extensive list. Anyone who's participated in a clinical trial can tell you how extensive the testing is that's performed on them. And so we're measuring, all of the things that we think we can do to measure impact. MRI, clinical outcome, and biomarkers.
And I think we're going to get the hook in just a minute. I will also add in the example of the Alzheimer's trial space, it's the duration. So with all of these neurodegenerative disease, you have damage building in the brain for a long time. And in the Alzheimer's space, it's a 35% slowing of the course of disease is what's been recorded with the lecanemab, for example. And that kind of thing isn't necessarily going to show up in 3 months. It's the course of seeing over a longer period of time, and it's identifying people early in the course of their disease so that it can slow and predict them. There's also symptomatic agents that are being tested. I know we focused on disease modifying today. AFTD is funding, for example, clinical trials through our Treat FTD program with the Alzheimer's Drug Discovery Foundation to try to get symptomatic treatments also to the market and to the families that need it. You've heard, I think, throughout today how complex the space is and how much research is underway, some of which is visible in the clinical trials that are desperately seeking participants and some of which is behind the scenes. And I also want to call out that every single company running a clinical trial is relying on the data that's been generated from the NIH-funded research. So reminding your politicians that bipartisan support for the NIH has been so essential for everything, for the progress is really important.